Good morning. Why don't we get started? We've shifted our agenda around a bit this morning, you may have noticed. So what we're going to do first here is do our business meeting. We're going to get the boring stuff out of the way first. We'll do that relatively quickly, and then we'll move on to our program, which is really meant to highlight our new president, Herman Haller, as well as our faculty. Oh, yes. It's always Jerry. Want to welcome everyone here today. Certainly, this is a day that we all look forward to. I certainly look forward to. The board looks forward to. We like visiting with the corporation, the faculty, our staff, the whole group. It's nice once a year for all of us to get together and share in the joy of what's been created here. To begin our business meeting, I need a motion to approve our minutes, previous minutes. Second. Any discussion? All in favor? With that, we'll move to the Secretary's report and Alan Miller. We'll just hand you that mic.
Mr. Chairman, my report is that there are currently 240 members of the corporation. That includes 19 full members, 32 associate members, 3 honorary members, 152 life full members, and 34 life associate members. 2 members of the corporation passed away this year. Life full member, Dr. Robert Wondergem, and life associate member, Peggy Forrester. That is my report, Mr. Chairman. Thank you.
Could I ask for a moment of silence for the 2 members who passed away this past year? Can I have a motion to approve the Secretary's report? Second. Any discussion? All in favor? Aye. Any opposed? Next, we'll do Ed Benz with the nominating committee.
Thank you, Peter. We have 2 slates of candidates to present to you. I can assure you that there was absolutely no collusion in coming up with these names. So to re-election for the Board of Trustees, and all of these members have agreed to serve for the class of 2022, Peter Allen, Alan Miller, John Hayes, Frank Homan, and Herman Haller as ex officio in his new role as president. You want me to do the whole slates and then approve them?
Sure.
Yeah. And then the second for your approval is nominees submitted for membership on the nominating committee. Edward Benz is chair, Barbara Belts, Jerry Bowers, Phoebe C. Boyer, Alan Miller, Wistar Morris, and Patricio Silva. Do I have a motion to approve? Yes, Jerry. Well, with apologies to Joanne Furstenberg also, could we get a motion to approve the amended slate? All in favor? Thank you.
Thanks, Ed. Tom Boyd will present the treasurer's report.
Good morning. I'm Tom Boyd. This is the Treasurer's report to the annual meeting. Let's go to the first slide, please. So this slide shows the statement of activities for the year 2017 in comparison with the prior year. Activities include operating support and revenues, as well as operating expenses. Looking at the first line of numbers, the operating support and revenues for 2017 were $10.5 million, marginally higher than the prior year. As shown in the second line, operating expenses were $12.2 million, or $350,000 higher than the prior year. The net of operating revenues and operating expenses resulted in a net negative change to assets of over $1.5 million. Moving down to the fourth line of the chart, showing non-operating items, grants and gifts for capital purposes were over $2 million in 2016, and then another $1.2 million in 2017. These amounts represented the use of the state of Maine bond funding that we received to construct the Maine Center for Biomedical Innovation and other important infrastructure projects at the lab. These building projects were completed on time and within budget in early 2017. On the fifth line of the chart, investment return for 2017 was a positive $731,000. Over the course of the year, the total net change, the total change to net assets was a positive $382,000, and this resulted in a balance of $25.8 million in total net assets at the end of 2017. Next slide, please. Is there a...
Keep pushing the button.
Oh, you do that? Please. Go ahead to the pie chart. There we go. Thank you. This chart illustrates how the institution covered $10.8 million in operating expenses in 2017. As you can see, 66% of funding for operations comes from federal grants, shown here in the blue and red sectors. 43% of the funding covers costs at the MDI Biological Laboratory, shown in the blue sector, and 23% of the federal funding, amounting to $2.5 million, is distributed to colleges and universities within the state through the IDEA networks of biomedical research excellence, or the INBRI grant, and that's shown in the red sector. Two federal grants from the National Institutes of Health provide major support to the institution. First, the INBRI grant provides $1.5 million to the institution and $2 million to subawards annually. The renewal of the INBRI grant was submitted in early 2018. Second, the Centers of Biomedical Research Excellence, or COBRI grant, provides $2.2 million in support of faculty annually. The renewal application was submitted last fall and was just awarded. Can we go to the next slide, please? This shows the financial position in terms of cash and investments. MDI Biological Laboratory remains in a strong cash position. Funds that were set aside for strategic growth are important to maintain. As you can see in the first line in the red box, cash decreased slightly from 2016 to 2017, but remained high, amounting to over $9.2 million. As shown in the third line down, investments gained even after being offset by the amount used to fund fellowships and programs. On the liability side, the institution continues to pay down the $5 million bond that helped finance the new laboratory building in 2007. Regular principal and interest payments are made. This is shown in the red box on the sixth line down. Over $4 million. Sorry. Is this better now? Over $4 million in debt remains to be paid off by the year 2036. Net assets here are shown broken out by unrestricted, temporarily restricted, and permanently restricted. As you can see, net assets totaled $25.8 million at year-end 2017, as shown in the red box. Can we go to the next slide, please? The $25.8 million in net assets are distributed among 43 different funds or categories. Some funds, like the General Operating Fund and plant assets, are unrestricted. Other funds are restricted for specific purposes, and the institution may use all or a portion of the investment income that comes from these endowment funds. All are tracked and accounted for separately by the lab. The strategic plan calls for growth in research faculty and education programs, and it also calls for diversification of revenue streams to support growth through varying funding climates. The board raised and set aside reserve funds in anticipation of the inevitable gap between the recruitment of new faculty and the funding of their research. To this end, $750,000 was pulled from reserve to bridge this gap in 2017. The cash balance of the reserve fund was just under $5.7 million at the end of the year, with another $600,000 posted in pledges receivable. In addition to the funds listed here, the Maine Community Foundation manages the Salisbury Cove Research Fund, established by Tom Marin and the Marin Foundation. Funds are made available each year, and we remain grateful for this support. This concludes the Treasurer's report for the annual meeting. Thank you.
Thanks, Tom. We have a motion to approve the treasurer's report. Second? Dan?
We've been looking at this diligently for the past several years. We had a discussion of it this week at the Finance Committee meeting. We plan to work on it actively over the next 2 years. We are running a deficit in operations, and it's quite noticeable, and we cover it by shifting funds from our reserve fund into operations to cover it. And we need to do that to cover our responsibilities to the bank for the bond that we hold, so that we maintain in a good financial position on paper. But we do run a deficit. It's part of our strategic plan to diversify our sources of revenue, and this is the thing that we need to work on in the next 2 to 3 years to solve. We got the COBRE grant approved, which is a major achievement for the lab, and I hope someone talks about it today. But that won't solve our problem, and our faculty has been spectacular at getting new grant support from the government in terms of independent grants, but that won't solve our operating deficit. We need to diversify our revenue streams with either philanthropy or new business relationships or other sources that we can find so that we have a stronger position. We can go like this for a few years, but we can't go indefinitely like this. Any other questions? Thank you.
Thanks, Tom. Hearing no further questions, all in favor? Any opposed? Next, we'll have the Audit Committee report from Max. Got the mic.
Difficult on one hand, of course.
One more. I'm holding it behind my back.
Okay. I am Max Brennickmeyer, the chairman of the Finance Audit Committee. The Audit Committee is made up of Spencer Irving, Hank Homan, Mr. Morris, and Dean Reed. We met on Tuesday, the 24th of 2018, to meet with the auditor, Annette Gold, of HMV LLC. That is a name change from their longer first, which was Horton McFarland Veazey LLC. Basically, everything has changed except the name. Everything is the same except the name, sorry. Annette Gould said there were no changes that she had to make to our numbers prepared by Claudine Lurvy and her team, and that the numbers for Mount Desert Island Biological Laboratory as of December 31st, 2017, and the changes in it of net assets and in cash flows for the year that it ended according with accounting principles generally accepted in the United States of America. It was a clean audit. It was the cleanest audit that we have seen for the years I've been here, and that's since 2003. So it was a very clean audit. Thank you, Claudine and your team.
Thank you. Thank you, Max. Can I have a motion to approve the audit committee report? Second? Any discussion? All in favor? Any opposed? With that, that concludes the business portion of the meeting. Is there any old and new business? Can I have a motion to adjourn? So moved. Second? Any discussion? All in favor? Great. So now we'll move into the fun stuff. You'll have to forgive me a moment because yesterday morning I was on a boat in the southern Bahamas with 4 connections to make to get to Bar Harbor. And it was all going swimmingly well until I got into JFK. And then that was a bit of a problem. The Bangor flight decided to be canceled at midnight. So I'm sorry that I was a little bit late this morning. And forgive me for any stumbles I may make due to lack of sleep. And that comes from someone as a surgeon who's sort of used to that kind of thing. So I would argue that if most research institutions, academic organizations, universities will consider one metric of their success to be peer-reviewed grant funding, and the highest level of that peer-reviewed grant funding is NIH grant funding. And I would argue that that's a very high metric, strong metric. Imagine if I argue this, someone in the audience will meet me at that door on the way out to say that it's not. However, if you look at all the rankings for medical schools or hospitals, when you look at even rankings for hospitals, what goes into that ranking, a lot of that is NIH-funded research and how well the faculty are funded, how much research is going on. And I would argue that if you look over the past few years, should I just use the handheld mic? Why don't I just try it out? Sure. I would argue that, can you hear me in the back? I would argue that if you look at MDI Biological Laboratory over the past 7, 8, 9 years, when we've made this transition to a full-time research organization, you will see a continual increase every year in the amount of funding, peer-reviewed funding that's coming into our organization. And if you accept that metric as a metric of success, I think that you have to argue that the laboratory has been very successful over these past years that we've transitioned into a full-time operation. And I would say that that metric has continued even over the past year. Since our last meeting here in 2017, July of 2017, our laboratory has continued to successfully grow and evolve into what is a nationally and internationally recognized research organization with this continued focus on regenerative biology and regenerative medicine. And our faculty over this past year have continued to be very impressive. Actually, I would say they've become quite a scientific force. And their accomplishments over the past year are many. We heard last summer about Wood's SBIR grant. This was funded just this past year. This allows $1.5 million in funding to go into Novo Biosciences to help for further development of this compound, 1936. Sciences has gained a lot of publicity around the state and even nationally. I think it allows us to have the recognition that we indeed can take our discoveries and try to shepherd them through this process of translation to see if these discoveries can be helpful to humans. There's a lot of excitement about this. I think this excitement has helped in many ways. I think it's helped our new building that was built on a Maine state bond for entrepreneurial innovation. This past year, the William Proctor Fund was established to provide our scientists funding to investigate some of these ideas, some of these discoveries. The NIH is not interested in risk, and a lot of these discoveries take a significant amount of risk in order to bring to their fruition, and Dwight's work is certainly indicative of that, and we're very hopeful that the SBIR grant will allow him to further develop this compound. This past year, Sam Beck joined our faculty, recruited to MDIBL on the COBRE grant. That was a critical recruit. The COBRE grant needs us to graduate our scientists and to have recruits to replace them. And we're extremely pleased that Sam joined us, and it allowed us really to put together, allowed Kevin to put together the COBRE grant, which was renewed. Last summer, you heard about Vicki Losick, who was named an outstanding investigator by the NIH. That designation just so happens to carry with it a $1.7 million grant award, which came through this year, and we were very pleased to see that come to fruition. That award also allowed Vicki to graduate from the COBRE, and that's what the COBRE program likes to see, is graduation and replacement growth. That award made it so that all of our investigators on the initial COBRE had graduated, and allowed for Kevin to put in a very strong application for renewal. In May of 2018, as you heard, the INBRE grant was renewed. This is a grant of $18.3 million. This grant is run out of MDIBL. There are awards throughout the state, and I actually believe throughout northern New England. I think there are some sites in New Hampshire as well. This grant allows us to provide core facilities and financial support for junior faculty. It also allows support for education of undergraduate and graduate students to teach them about biomedical research, biological research, hopefully to train Maine and New Hampshire residents in these areas so that we can further the biomedical research institution effort in this state. And not to be outdone, in June of this year, we were notified of successful renewal of the COBRE grant. This is has an enormous impact on our budget, as Tom just showed. It is a testament to Kevin's success in managing that grant and his grantsmanship in putting it back in. These are very difficult grants to have renewed. They were even more difficult to have renewed this year, and we owe Kevin a real thanks and tribute to his work in getting that grant completed and successfully through the process. Not only did that require all the faculty's hard work and all of the and Kevin's hard work, but also all of the staff here at the laboratory put in a tremendous amount of work this year to see, to shepherd all of these grants through the process so that we could maintain our financial status. We're a small place, as everybody in this room knows. The staff here work tirelessly. Claudine, Jerry, Judy, Jane, both in her scientific efforts as well as leading our educational work, and Mark just do a truly outstanding job in keeping this place running. And when we're this small and you're one person in one division, you need to be able to collaborate with the others and work together. And the thing I've been impressed with the most is how this group and our senior staff and all staff on this campus have really worked together to complete the mission that we've set forth to them. This past year has also been one of transition. As we all know, the board spent a great deal of time and energy in identifying Kevin's successor. Ed led the search committee, and this committee spent time over the past 1.5 years identifying our next president. And this was a national search, really trying to focus on this area of regenerative medicine. As is often the case, after looking far and wide, we found the person with the greatest scientific talent, the person with the most leadership experience, the person with the most extensive experience with industry, and the person with the greatest understanding of our institution right here on our own campus. How often does that happen? All the time. And we're extremely pleased to announce that Dr. Herman Haller has agreed to be our next president. Dr. Haller received his medical degree at the University of Berlin. He did his postdoctoral work at Yale and has been professor and director of the Department of Nephrology at the Hanover Medical School in Germany since the 1990s. Without a doubt, Dr. Haller is an international thought leader in kidney disease, hypertension, as well as kidney and blood vessel regeneration. He has been an investigator here at MDIBL for over 2 decades, and he is an incredibly accomplished scientist. He has over 700 peer-reviewed publications on his CV. He holds 6 international patents. He has founded 4 biotech companies, and he sits on many for-profit and not-for-profit boards. He has run a very large laboratory in Germany for many years and has been very successful in moving that laboratory forward as a group. I think that he is someone who does very well in mentoring and bringing his scientific group together, and we're very pleased that he is our next president of MDI Biological Laboratory. We're glad that Herman and his wife Ude are here with us, and I'm going to turn the podium over to Dr. Haller. Please join me in welcoming.
Don't know what to say, I mean, I just have to recover from this very friendly introduction and have to count all these numbers, whether they are true. Members of the corporation, colleagues.
Yes, that is muted right now. There you go.
That's much better. Okay. Members of the corporation, colleagues, dear friends, it is an honor and a challenge to stand here and discuss with you the future of MDIBL. I will not talk about the honor. I mean, all these faces looking at me, and some of them previous directors looking here from the floor, doesn't make my job much easier. I will talk about the challenges. And talking about the challenges, you have already heard Dave Dawson asked about where is this deficit coming from, and that will be a challenge. We've heard about the successes. I will not talk about the problems, but I will talk about how to do that. You look at that and you see that I'm a builder. I'm not tearing down things and then make something new, and I will not give you a structure of something very novel, but I'd like to build the lab from the existing parts which we have here. And these parts are, and you have listened to Peter, they are just great. We have the buildings here, the new buildings. Over the last couple of years, this place has changed so dramatically that some of you coming here and have not seen the development over the last couple of years, they get lost on campus. So yes, we are small, but nonetheless, we need a map. We now have a faculty. We have a faculty which is outstanding, and you've heard that. It's small, but it's focused, and so it's actually fun with all these parts together with the faculty and the staff to build the MDIBL for the future. One caveat. I will not tell you that I understand everything. I've been here for 20 years, but I'm learning. I've been here now for 2.5 weeks, and I can tell you my learning curve is steep. And sometimes faculty has to remind me, well, you said something different, change that. And yes, we can do that together. I have ideas, you will see that, and sometimes we have to discuss things, but we want to do this together. Now, as every good architect can tell you, you start with the roof. You know? And for a structure for the future, the first question is really, after all these changes over the last years, what are we here for and why are we necessary? I will not go into the 1900s. Then it was very clear this place was to come and investigate marine animals. And this is why we had all the buildings here, because it was too hard to transport them to Boston or to other places, so they made a laboratory at the sea. This is not the case anymore. And we have started that. Some of you have been dramatically involved when we started a year-round faculty. So it becomes even more important, are we now a small Harvard? Or are we still a marine biology lab? Definitely no. What are we and why are we necessary? We have come a long way. And I think this has to go into the equation. I mean, you see here different people. You see here the lab as it was. You see the new building. This is almost 20 years now. You see faculty and you see more new buildings here. So the question is really, what is our mission? And I can assure you, I don't like, and we have already started talking about this, I don't like meaningless sentences. And very often, missions are like that. You know, you can read it or you don't read it. So the real question is, what are we doing here? And the lab has been famous as a meeting place. I like this here. You know, this is a group of researchers in 1940. And you know what they are discussing? They are discussing regeneration. They are discussing cell growth. I was astonished that at that time there were people meeting here not discussing the kidney or some glomerular function or tubular activity, but they were discussing growth of tissue and cells. So the lab has been known for interesting hypotheses, for excellent science, for heated discussions. This is the place where you can actually challenge other people, because the next day you can come back and say, well, I'm sorry, I didn't mean it like that, but you're still wrong. It's about enthusiastic education. A lot of students left the lab, and they either became scientists or medical doctors, but a lot of them didn't, but they remember the lab. We have people coming here after 10, 15 years because they experienced a place where education was important. It's a place for thought and novel ideas. And John Epstein said to me, you know, this place has been known for career-changing ideas. So actually you need a place, and this is an academic campus. You see the Harvard, but there people are too busy. And we are busy here. I mean, the faculty and me, we are running around all the time. But what we are really famous for is that we are a campus where you can have all these things, and I think that's the roof. We are an academic campus. Now, the question is, how do we support the roof? And we have two pillars here. And the one is education. And we're doing great with education, but, you know, schools are always underfunded. And we have the faculty, and we'll talk about this in a moment. So we are doing great with grants. But when we started a year-round program, a grant paid for 80% plus. And these days, it's only paying for 50%. So this means for every grant we get, we have to generate another 50% from somewhere else. And if we want to do this, we have to broaden our foundation. And these are the 4 areas, in modern times I would say business units. These are the 4 pillars which I think should carry this route. And that's the faculty, a fellows program, education, and last not least, very difficult, biotechnology. And because we wanted to demonstrate you how strong we are, and I think you need to understand really what's going on here this year, that the faculty, and this is our faculty, that the research groups where the actual science is done at the moment, not everybody, but a smaller group of the faculty will present in a very, hopefully, very precise way what they are doing. We will not have questions and answers. Hopefully you will have questions, but what we will do is, at the luncheon, there will be tables, and each of the scientists will sit at one table, and if you remember your questions, you can go to that table and sit down and ask these questions. So we will just present. We'll go through this in a hurry, not to be too long. And then after that, I will go also rapidly through the other 3 pillars. And now we start in an alphabetical fashion with Sam Beck.
Good morning. My name is Sam Beck, and I am a computational and cell biologist in MDI Biolab. And it is a great honor to introduce my study in this meeting. And we study about the human aging with using the disease model called premature aging syndrome. Let me show you 2 examples of the premature aging syndrome. The first model is called progeria, or HGPS. And this picture shows the progeria patient at their age of 1, and they look apparently normal. And when they become 2 years old, they start to age faster, about 7 times faster than normal kids. And here is another example called Werner syndrome. These patients are at their age of 13, 15, and 21. Apparently, they look normal, but after some time point, especially after the puberty, they start to age faster. And at their age of 48, they look like 70s or 80s. And the molecular cause of these diseases are quite well characterized. The progeria is caused by mutation in lamin A gene, and lamin A is the protein, the structural protein that are located at the periphery of the nucleus. And this mutation causes the disruption of the morphology of the nucleus. And on the other hand, the Werner syndrome is caused by the mutation in WRN gene, and WRN protein is involved in the DNA damage repair. And one question that we have about these two diseases is how these totally different mutations can cause similar phenotypic outcome of the premature aging. And regarding this question, probably the answer lies in this structure. This is the schematic drawing of the nuclear periphery and in normal young nucleus. And the nuclear envelope separates the cytoplasm and nucleus portion of the cell. And as you can see, inside the nuclear envelope or the periphery of the nucleus, we can see very specialized layers of the protein and chromatin. And the first one is the INM protein, inner nuclear membrane protein. These proteins are transmembrane protein that are associated with inner nuclear envelope, and they interact with the fibrous network protein called nuclear lamina. And this nuclear lamina further interact with heterochromatin portion of the chromosome. Heterochromatin is the condensed form of the chromatin, and this, as a result of this series of the interaction, this heterochromatin localized toward the nuclear periphery, and they are spatially segregated from its counterpart, euchromatin, located at the nuclear center. And one of the interesting things about the aging is that aging leads to the disruption of all these peripheral structures. Here are some examples. There are multiple aging contexts, including the premature aging disease, progeria, and Werner syndrome. And cellular senescence is known to be enriched within the normal aged tissue. Although their causes being different, all they show some level of the disruption in INM proteins and nuclear lamina layer, and all they show the similar decondensation of the heterochromatin structures. And these structural changes with age is very well known, but we still don't know how these changes can contribute to the degenerative changes with age. Regarding this question, our previous study suggests that the answer to this question lies within this very specialized DNA element called CpG island. The CpG island is the mammalian DNA element observed in human and mammalian genome, and this element has a higher frequency of the CG dinucleotide compared to the other regions. And those elements are usually associated with promoters, and as a result, about 60% of total gene contains such a structure, and we call them CJ positive gene. And the other 40% of genes that do not have such structures are called as CJ risk genes. And most of the housekeeping genes are CJ positive genes, and CJ risk genes are generally expressed in a very tissue-limited manner. And we tried to understand how these two types of genes are differently regulated, and we performed a large-scale computational analysis. And long story short, what we found was this. Basically, CJ risk genes are regulated by interconversion between euchromatin and heterochromatin, but CJ positive genes remain as euchromatin even when they are inactivated. And we also found that the CJ positive and risk genes have totally different nuclear localization pattern. Basically, CJ risk gene localized toward the nuclear periphery when they are silent, forming heterochromatin, and their activation coincides with their extrusion into the nuclear center. But for the case of the CJ positive genes, they remain at the nuclear center regardless of their transcriptional activity, even when they are silent.
I think.
I have to be fast.
75.
Oh, you counted.
So how do I get this up here? I need help. I'm technologically challenged. There we go.
I'll do this for you.
I can get it. Good morning, everybody. So I'm just going to tell you about what we're doing in my lab. What we're very interested in is, can everybody hear me? Okay. What we're interested in is chronic stress and the question of how does chronic stress undermine health and accelerate aging. So we all experience stress. Stress is increasing in the world. Chronic stress is a problem. We experience it throughout our lifetime, beginning from the time even before we're born. As you get older, stress contributes to diseases of aging, but then those diseases of aging are also stressful. So you have kind of a bi-directional relationship as you get older. Early on, the stress is more from the outside. I'm a developmental biologist, so I'm interested in the origins of things. So what we're kind of looking at is the stress from the very earliest stages of life and development, what happens prenatally in utero, if in chronically stressed mothers, such as might occur in mothers who are under extreme poverty or in abusive situations, war zones, and so forth. So Herman asked me to address 4 questions in my talk. Why is my research important? Well, chronic stress is a major factor contributing to degenerative disease and health decline with age. We know that for a fact. Chronic stress early in life increases the risk of developing inflammatory diseases later in life, all kinds of diseases that are environmentally induced or stress-induced, asthma, arthritis, cancer, psychopathology. So the biological mechanisms through which chronic stress undermines health are really poorly understood. So that's what we're trying to understand. What is known is that chronic stress leads to chronically elevated levels of cortisol. It's a steroid hormone that regulates many processes and much of the physiology of our body, and including inflammation. So exposure to chronically elevated cortisol during very early life, for example, in utero as you're developing, in a mother who is chronically stressed, we know that has long-term developmental effects that undermine health. That's the buzzword for that is developmental programming. So our hypothesis is that cortisol, it's a biologically active steroid hormone, cortisol-induced developmental programming has persistent effects throughout the life on stress-responsive gene regulation. So that's our hypothesis. How are we going to address this? Well, we use zebrafish as a model organism. Zebrafish is a great vertebrate model organism. It has a stress system very much like our own. We use cortisol as a stress hormone. So we treat zebrafish embryos with cortisol. We just put it in the water as they're developing for the first 5 days of development. And then we see what happens during the treatment period, but we also follow the fish up into adulthood to see how that treatment during development affects their adult physiology. And then we are also taking them into subsequent generations to see if there are long-term intergenerational effects. We can take blood cells out of the fish without killing the fish and culture them ex vivo and see what kinds of effects there are on the cells themselves. So we have a publication that came out 2 years ago that describes some of the initial results that suggest that this is a good model for looking at this. The fish have long-term effects into adulthood. So the tools we use, zebrafish are great because you can get genetically modified zebrafish, for example, that have a green fluorescent protein. This strain has a green fluorescent protein that's driven by a glucocorticoid receptor, so it responds to stress or to cortisol. You can see these fish here, these larvae that were incubated with cortisol are glowing more brightly green than the ones that were controlled. We also have zebrafish lines that have the cells of the innate immune system, neutrophils and macrophages, which are the cells that mount the inflammatory response. We can actually watch them mount an inflammatory response in real time. So we have these kinds of tools in the zebrafish. We also are doing a lot of high-throughput DNA sequencing. We have a great bioinformatics core here at the MBI Biological Laboratory, and we can look at global functional genomic effects of this cortisol exposure. So finally, how will my results change the world? Well, I don't know. But I'll tell you that they will lead to a better understanding of how chronic stress undermines health. And also, glucocorticoids are very widely prescribed drugs. Synthetic glucocorticoids are used as anti-inflammatories for all kinds of conditions. They have horrible side effects if you use them chronically. They lead to obesity, metabolic syndrome, all kinds of bad things. But they're very effective as anti-inflammatory drugs. So we're trying to understand how the dynamics of gene regulation work with these glucocorticoids, and it could potentially, what we find could potentially lead to development of better drugs for treatment of inflammatory disease. So I'll be happy to tell you more about it at lunch.
Well, if the faculty doesn't need a microphone, if you can understand me, I don't think I need a microphone either. Now, James Goodwin, I'm not going to present his slides. He is stuck in Munich. At the moment he's flying, but he didn't have a visa. He's Australian, coming from a meeting in Vienna. So he has been now for a week in Munich. I am asking him whether he likes Bavarian beer. He tells me he's too stressed to drink that. He'll present his data himself at another occasion. But I think it gives you an impression how complex it is what we're doing. When you look here, you see new animals, you see limb generation, and it will be fascinating to hear him talk next time. Now, this is me. Or this is us, Maria Schiffer and I. We have been running here a lab for the last 20 years. Thankfully, you have already heard all that. But this is important. I like translational research and have done basic research in endothelial cells and vascular biology since my postdoc. But I'm also involved in drug development and clinical trials phase 1 to 3, and I hope I can bring this here, not only with my research group, to the table when we develop MDIDR. Now, what are we going to do? Our problem is that the research group, Mari has been very essential. He's becoming head of nephrology at Erlangen University, and I hope he'll be back as an independent researcher next year. And here you can see our development over the last 20 years in the lab. The research topic is to understand the vascularization of renal tissue, or how do blood vessels get into tissue. We can now nicely, from stem cells, make chunks of kidneys. But these chunks of tissue are not vascularized. So the question is really, we have vessels developing within these chunks of renal tissue, but then at the same time we have to have vascularization coming from the outside, and this is completely not understood. This is like these two hands touching and then life starts. That's exactly what we'd like to know. Which growth factors, which matrix molecules, what is the driving force on both sides, and what brings them together. We use 3 different approaches. First, we use the zebrafish you have already heard about. We can make transgenic zebrafish, and we can have all sorts of colors for development of blood vessels, for growth factors, for other factors which are involved here. I mean, we can actually look how the blood vessels come from the aorta into the kidney and how they come or they develop within the kidney. We have laser-based microperfusion systems where we can use stem cells and we grow a capillary network under flowing conditions, and there we can test in vitro whether the molecules we have identified here actually work there. And then we will use organoids where we can differentiate within a chamber renal tissue and see, and here you can see some of the cells are endothelial cells, how they develop. How do we improve the world? We want to make a new kidney. I mean, if we understand how we can vascularize the tissue, then we can actually grow these chunks of tissue, and then we can vascularize it. At the moment, that's not possible. We have to implant it into animals, other animals, and then we have an irregular vascularization. So we have here also a laser-based chamber where we mimic matrix with novel substances. We have an aorta here, we have a ureter here, and in between we try to differentiate, use growth factor gradients and oxygen consumption and metabolic changes to understand this and make a complex tissue. And then in the end, hopefully, we have a vascularized organ. The NIH has picked this up, not our work, but they don't want to spend $4 billion per year on dialysis anymore. And they said, well, we'll take this money and seriously, we want to have a new kidney. So there is a NIH program, Kidney X, and together with other people in the state of Maine, in Portland, there are people working on that. We try to achieve that. Now I have to disappoint you. In the program, you see intermission, but we are so fast that I think you'll skip intermission, and we'll have the next presentation. The next presentation is by Vicky Losig, and I promise you we will be as fast as possible to get you to lunch.
Oh yes. Do I need the mic? Or talk loud now? Okay. It's a pleasure to be back here. It's on. It's good. Okay. And I did talk last year, but I'll just give you the short version this year. Okay. So my group is, interested. Our main goal is to improve our ability to heal, and that's because we have a lifetime of tissue damage, whether that be a small wound that you get with maybe a paper cut, or something more serious like a heart attack. And in order to understand how to improve our ability to heal, we need to first understand how our tissues go about that. And you have damage to a tissue, and there's two mechanisms. One, we need the key step in repair is to replace what's been lost, and one mechanism is to do this by cell division. Okay? And the other strategy at play is for cells to grow in size to repair that wound. When a cell grows in size, it boosts its DNA content by making extra copies of the genome, and that is known as a polyploid cell. I try not to use jargony things, but I have to use this word. Okay? Polyploid cell. And, last year I gave this example, and I wanted to give it again, but you may not realize how common these big polyploid cells are. They are important in growth of many things, in particular something we love, the strawberry. Okay? And the way our strawberries grow bigger, you go out and you're like, oh, I got this little puny strawberry, but what you really want is this big one. And the ways our strawberries are growing bigger is by making bigger cells. This strawberry is made up of bigger polyploid cells, and that's why it's larger. And last year I told you we're in search of Maine's polyploid strawberries. This is an updated photo of my kids, and we're still looking for them. We got a lot. They always go for the biggest ones. Okay? So not only are our strawberries growing by making these big cells, but also our tissues are repairing by making big cells. And these are just a few examples here. The liver, the most regenerative organ in our body, it regenerates by making large polyploid cells. And then recently, an example that comes from the zebrafish in the heart, this is from Ken Pasta's lab, a visiting scientist here, a longtime visiting scientist, showed that in the zebrafish heart, which can regenerate, also forms big polyploid cells during repair of its tissue. And my lab works with the fruit fly, and we show that these polyploid cells also form in the fruit fly. So we want to discover how polyploid cells help to heal our tissues. And how are we going to do this? This is my dream team to success. Okay? We're going to use the fruit fly, which, unlike mice and zebrafish, has we can generate mutants really fast. It's really cheap to work with in the lab. So it's an excellent model system, and it's ideal for genetic and cellular analysis. We can make mutants really quickly. And we can see all the cells are changing, so we can see the formation of these big polyploid cells. We puncture the flies, we see these cells form, and then we can identify the genes that are critical in making these big cells. And this work is all being done by several dedicated scientists that I was able to recruit here. They're all graduates of universities in Maine or soon to be. And I've been able to convince a young group of excited scientists to take on this problem and discover the role of polyploidy in wound repair. And of course, I wouldn't be able to do all of this amazing work without the support of Mount Desert Island, as well as the laboratory, all the staff here, and funding. And lastly, how is this going to change the world? Well, I would like for people to realize that what can polyploidy do for you? It grows your strawberries bigger. It helps to heal our wounds, okay? And we're not going to be talking about stem cells anymore. We're talking about the importance of polyploid cells, okay? Polyploidy is the remedy. And we are having a symposium, the first of its kind, to focus on this topic, bringing experts in the field that work on polyploidy and organ development and repair and disease. It's happening this fall. If you want to learn more, you can look at the website here. I encourage you to come if you're interested in hearing about this topic. It's really incredible. We have scientists that are so excited about this meeting, they're willing to come over from Japan, as far away as Japan. So it's really an incredible opportunity for us to learn more about the importance of polyploidy in growing our cells, developing our organs, repairing our tissues, and how it can function disease. With that, I'll leave it, and you can come to my table and hear more.
Okay, Eric? I think you already are overwhelmed by the quality and what we actually do here, and there is much more to come.
Hi there, I'm Eric Rogers. Let's see if I can. So we were asked to put together a very short presentation focusing on 4 questions. One of them was, why are you doing what you do? The reason that we look at aging isn't just because we romanticize about the old ideas in the past about a fountain of youth or plants with, that would yield eternal life. It's actually because now in the last 20 to 30 years we have a lot of evidence that aging is very malleable. It's not an absolute every time that you're going to age the same way, and actually you can change the rate of aging just with lifestyle changes. And so the whole idea behind aging, why does it work? Why does it work? Why would nature make a program where you could go in and mutate, literally change the smallest little part of a gene to slightly change its function? That small of a change, and you could increase lifespan tenfold. Why would Mother Nature have that program? Why does Mother Nature care? Well, she actually doesn't, is the truth. And so what happens is we have this theory, it's been around since the 50s. It's a theory of trade-offs, is the easy way to look at it. Antagonistic pleiotropy is the kind of fancy word, but essentially all it is the genes that are really important for you to grow and develop and reproduce. They can actually hasten age-related decline during adulthood. So the genes you need, the genes that are important and good, actually can become a bit of your enemy later in life. And this has to do with a very basic fundamental rule in nature. What does nature care about? What does a species care about? What does Mother Nature want? You have to be able to grow and reproduce, otherwise you're not going anywhere as a species. That's number one. The second thing, you have to be able to conserve resources when conditions are not good for growth and reproduction. That's what Mother Nature cares about. So there's these programs, we'll call them stress response programs, adaptation. Okay? And it's meant to allow an animal, from a worm to a human, to survive long enough, okay, to find the resources that they need to go forth and grow and reproduce. The good thing is, even after we're done growing and reproducing, we can tap into these pathways that nature has planted for us. And when we age, one of the things that's happening is we're losing that ability to adapt at the cellular level, the molecular level. Anything you can do to strengthen that is going to be an anti-aging strategy. Why is exercise good for you? What do you think you're doing? You're actually causing immense stress on the body. But it's a type of stress that we, from an evolutionary perspective, are used to dealing with. And it actually can have very positive consequences. You could exercise yourself to death, just like you could go sit in a sauna and kill yourself that way. But a small amount of stress that you can fully recover from is actually very beneficial. It's an idea, in the past it was known as hormesis. Now we know a lot more. So I study in the organism that really kicked off the revolution in our understanding of aging. And this is the tiny roundworm C. elegans up here. Why is this such a useful system even now for biomedical research? Well, it looks a lot different than us. It doesn't have a backbone. And as simple as that organism is, it has less than 1,000 somatic cells, non-reproductive cells, during adulthood. But it's got complex tissues and hormone signaling. It's got neurons. It's got muscle cells. It's got intestinal cells. It's also got an extremely rapid life cycle. So from an egg to an egg-laying adult in 3 days to death in 3 weeks. That's a pretty rapid life cycle. So anything we do to affect its health, to affect its aging, we can get our answers right away. If I get a wild hair in the lab and I say, you know, I think the neural cells are signaling somehow to the muscle cells, I can go and set up an experiment, cross some strains together, and in a few weeks, start that experiment. You want to do something like that in a mouse, you're looking at many years and many millions of dollars. So there are some things that we can begin to interrogate and begin to question very quickly. It's also a clonal organism. It's genetically very well characterized. Every cell is mapped, every cell. It's now used in high-throughput drug discovery. Imagine, rather than tissue culture in a high-throughput assay, you have a whole intact animal. These things, by the way, that is not actual size. They're 1 millimeter long, okay, full grown. So this is a microscopic organism. It's transparent. So all those pretty pictures people were showing earlier with fluorescent things, all that stuff has usually been extracted out, or it's grown in a dish. We can actually look inside this organism while it's still alive, while its metabolism is still functioning. You might think, well, God, how do you dissect tissues out of an animal that's only 1 millimeter long? Dustin Updike does it. He can take out the reproductive system. For me, I'm not that skilled, and it would be really hard to dissect neurons or something out of this thing. Fortunately, we have ways of combining genetics and biochemistry, so we can go in and biochemically isolate whatever we want from a specific tissue to get at answers for what function does this tissue have. So finally, the mantra in molecular biology is DNA, RNA, protein. Okay, that's your biology lesson for the day. We have the genetic code for all of the proteins of life. They get transcribed, so the code gets transcribed, and then it gets brought over to machinery that translates that transcript into a functional protein. We know a lot about this as a form of regulating how active our genes are. Much more recently have we really gotten to take a close look at just how selective genes are being active or not at this level of protein synthesis. And so one of the main projects we have, Jared Rollins was a postdoc in the lab. We're now finishing his projects through a collaboration on how dietary restriction works. Dietary restriction is one of these interventions that taps into those adaptive mechanisms and keeps you healthy and strong. But there's a lot of paradoxes. How is it a good thing to starve yourself? I mean, that it's so anti-intuitive from the way that I grew up, where, oh, we got to make sure I'm getting my vitamins and minerals and this and that and the other. So I can't get into all of the details of this, because even when I'm describing it to some scientists, their eyes begin to cross. So I'm just going to say here that what we discovered after years of intensive study of many different processes, we've made, I think, a major discovery. And that is, there's a quality control, something called a quality control program for this stage in the cell. It's actually not just a quality control program we have found. This is actually a way of regulating genes that are really important for adapting to that condition and really important for getting the health benefits out of it. Just briefly say it's called NMD, nonsense-mediated decay, actually alternative splicing in nonsense-mediated decay. But the NIA in particular was very excited about this idea. They awarded us an R21. When we first got our score back, we were at the 13th percentile. The funding was 11. But I actually heard something from the program officer I never thought I'd hear them say, just because they're usually very cautious about making any kinds of promises. She said, if this goes through again and it doesn't get funded, this is actually something we here at the NIA are interested in you doing. It's very novel and has a high potential payoff. And that's what that grant, the mechanism of that grant was. The other very provocative line of research that's become very provocative recently to me is how when you lower protein synthesis, which happens when you're dietarily restricted, okay, if you're energy restricted, you begin to synthesize less proteins, and you also begin self-eating. You begin eating your own cells. So how do you maintain tissues when you're not replenishing and you're just eating? And especially, what if you're an animal that needs to find food? You need to be able to hunt for the food. So your muscle, your skeletal muscle, your body muscle has to be maintained. So there's a balancing act. The body has to sacrifice something and preferentially maintain certain tissues while at the same time conserving. And one of the things we found is that if we squeeze protein synthesis the same way it's squeezed during dietary restriction, just through neurons or just in the reproductive system, an amazing thing happens. We actually see myogenesis. We see the formation of a myogenic, activation of a myogenic program that goes to restoring. And the root is actually through RNA. So your protein synthesis ends up getting turned down. One thing you can do if you've got a really important structural component you want to reinforce is flood the system with this part. Now, what happens when we do that without dietarily restricting? Could we actually reverse a natural tendency as we get older, which is muscle wasting and sarcopenia? It's a huge problem. So I'm very excited. This work was started by a postdoc in the lab, Amber Howard, and now Santina has taken it to, Santina Snow is a technician in the lab. She's taken it to a whole new level with this discovery, and of course now she's off to grad school. So we wish her the best, but whoever gets her is gonna be a very fortunate PI. And with that, I'll say thanks for listening.
Well, Eric's already introduced me to some extent, but I'm Jared Rollins, one of the newest faculty here at MDI Biological Laboratory. And essentially my lab is interested in extending healthy lifespan in humans. And we do that by studying dietary restriction. So why do we study dietary restriction and translation? Well, we study dietary restriction because it is the most robust intervention that we know of that extends longevity. And it extends longevity in virtually every organism it's ever been tested in, whether it's a simple organism such as yeast or a more complex organism like the worm that we study, mice, and there's even humans that are practicing dietary restriction to see if it will extend their health and longevity. We don't know if it extends the longevity in humans yet, but it does have health benefits, and we'll hopefully find out. Now, we know as scientists that one of the ways that dietary restriction works is by changing the genes that are expressed. And as Eric already alluded to, that gene expression is complicated, but we can essentially break it down into two steps. Transcription, where DNA is made copies of that are termed RNA, and then translation, where this RNA acts as a template to make a protein. And a lot of the research has been focused on the changes that occur transcriptionally under dietary restriction. And that's been phenomenal in giving us an idea of the importance of insulin and insulin signaling and targeted rapamycin signaling pathways and actually extending longevity. However, this other area of research, right, the regulation that occurs translationally has been largely ignored, in part because we just didn't have the techniques to do so. And part of my research actually with Eric was looking at what is the translational regulation that occurs under dietary restriction. And what we saw is that there's a whole multitude of genes that are already known from other research to have a role in longevity that are being regulated primarily at the level of translation. Some of these anti-longevities are down under dietary restriction and some are up. And so I'm interested in understanding what is this mechanism that allows this translation, this regulation to occur under dietary restriction. And that led me to this hypothesis that our lab is currently investigating, is does the ribosome, right, the molecular machine responsible for translation in the cell that all organisms share, does the ribosome regulate aging by selecting which genes are translated? And this hypothesis is based on 3 key observations made by ourselves or in the literature. One I just talked about is that many longevity genes are regulated almost exclusively on the level of translation, right? The other observation has been seen in yeast and mouse and hasn't really been explored yet in the worm, which is that there are different compositions of the ribosome, and these different flavors, let's say, of the ribosome actually help select which genes are translated and are present in the cell to be active. And then finally, we also have evidence in our data set that the composition of the ribosome itself changes under dietary restriction. So that leads us to one of the models that I'm testing, which is that under well-fed conditions, you might have one part of this molecular machine, ribosomal protein, is more abundant. And so that there's more of this flavor of the ribosome. And then this ribosome is going to prefer to translate genes that have no effect on longevity or possibly are translating anti-longevity genes. However, under dietary restriction, also called caloric restriction, you can have a switch in which of these ribosomal proteins are being expressed. And then that will change the composition of the ribosome, and the ribosome will then prefer to translate some of these pro-longevity genes and have the pro-longevity effect of dietary restriction. Some of the tools that we're using to understand and tease apart this mechanism, one's already been introduced, C. elegans, and one of the reasons that we use this, not only because of its short lifespan and its similarity to humans, is that it's got this wonderful genetic toolkit. So we can go in and essentially turn up or turn down some of these ribosomal subunits, ribosomal proteins that are changed under DR, and then ask the question, are they responsible for this, for the translation effect that we see on these longevity genes? We also use a technique called polysome profiling to essentially separate out the actively translated genes from the inactive genes. And then we can submit all those pools of genes to next generation sequencing and allow us to, in an unbiased, very broad way, ask, okay, what are the genes that are being up or down regulated, and are they being regulated translationally? We're also going to be using a proteomics approach, and that's a way of identifying the proteins that are present in a sample so that we can confirm that these changes in the ribosome composition are actually occurring under DR, and we're taking it a step further to see how the ribosome changes with age. So the outlook, how might this research change the world? Well, one, if we understand, right, how different compositions of the ribosome change how genes are expressed, that will essentially help us break this ribosomal code and can fundamentally change our understanding of how genes are regulated. But a broader picture, right, if we if we understand new pathways at how longevity is regulated, that gives us new druggable targets. And in the very big picture, if we find a way to consistently slow aging, not only will we live longer, but you slow aging, you'll also slow the progression of age-related disease. So instead of just treating all of these different diseases like atherosclerosis, cancer, diabetes separately, instead of treating them that way, we treat aging, and then we'll simultaneously slow the progression of some of these massive diseases that affect the majority of people here in the United States. And with that, I look forward to your questions at lunch.
Jared, I'm afraid, you will only have people at your table with nothing on their plates or the question, is that okay for my ribosomes?
I'm Dustin Updike. I'm one of the human control groups for those not practicing dietary restriction. So for those who I haven't met yet, I came here in 2012, and I was on the first phase of the COBRI for a couple of years, and now have funding through the NIH, through NIGMS. And for the past year and a half, I've been assisting the PI of the IMBRI grant, Dr. Jim Kaufman, on some duties with this grant that is a catalyst for biomedical research across the state of Maine. So before I get into my research, I just want to introduce you to the people doing it in the lab. And if you can find these guys at lunch or sometime and ask them what their contribution to the project is, that would be fantastic. So we have Dr. Elizabeth Marnick, who is a postdoc in my lab, Jesse Rochester, a graduate student, Catherine Sharp, a research assistant, Heath Fuquay is a undergraduate technician from the College of Atlantic. And then also with us this summer, Sarah Hallbrook is a graduate student who's rotating in the lab. Emily Shu is a undergraduate from the College of William and Mary that has come to us with all kinds of nematode experience. And then also with us is Esther Yoon and Haemin Min, who have come to us as graduate students from a collaborator lab in South Korea, and they'll be here for the next month. Now, as we know, most of us here have this focus on regenerative biology, and we use a comparative approach to look at this. And I just want you to look at this phylogenetic tree to realize how well animals regenerate. So this is metazoans, multicellular animals, and those highlighted in yellow have the extensive capabilities of regeneration. And some of them can regenerate the entire animal, the whole organism. Some of these are used in labs like planarians and hydra and nematostella. And then other animals such as these more complex animals, zebrafish and axolotl that James Godwin studied, have this ability to regenerate after wound-induced induction to regrow and regenerate limbs. So taking this comparative biology approach, we want to ask, what do these regenerating animals have that these other clades that do not regenerate, or where regeneration is extremely limited, what makes that difference? And this is where we come in, and ask, one of those differences is that the somatic stem cells in regenerative tissue express what we call germ granule components. And I put a little star next to all of these animals where germ granule components are found in somatic regenerative tissues. So what are germ granules? They're called germ granules because they're normally expressed in the germline of all of these animals. And the germline is our reproductive precursor cells, the cells that are going to turn into sperm and egg and give rise to the next generation. They are the key to understanding regeneration. They're found in the germ cells of all metazoans. They're this heterogeneous mix of RNA and protein, and they are sites of small piRNA biogenesis, and their core components are conserved across all of these animals. So these got messed up a little bit, but the VASA helicase, the Pee-wee Argonaut protein, and tutor domain containing proteins are found in all of these animals in these germ granules. So what are they doing there, and how are they promoting regeneration? Well, we're studying germ granule function in living, transparent, and genetically amenable animals. These are pictures from C. elegans. And we can use this system to ask what germ granules are doing, their dynamics, what they're comprised of. We can use CRISPR technology to put fluorescent tags on living worms and see how these granules segregate. We can use it to do bioinformatics and immunoprecipitate and look at protein interactions. We can do things like look at how germ granules are assembled and what we can do to disassemble them or to make them kind of disperse and go away. We also use a system to look at the genetics of germ granules. So what happens when we take away germ granules? What's the effect? And just the short story there is that you take it away and your germ cells start to turn into things like neurons. They don't no longer have that pluripotent or regenerative potential. We can then use CRISPR to introduce point mutations in germ granule proteins and look at how that affects the distribution or the aggregate formation of these germ granules and which components are impacted and how these germ granules are assembled. So Dr. Haller asked us how our research is going to change the world, and just shortly, the take-home from us is that these germ granules promote pluripotency, okay? And we think that we can address two very important questions in the distant future. And that is, can nucleating somatic germ granules induce pluripotency and regeneration? Can we actually use this as a tool to get pluripotent cells when we need them? And the other thing is that some of these germ granule components are expressed in cancers. So for example, melanoma, 70% of melanomas have germ granule components expressed in them. And 40% of breast cancers, the same thing. Can we use these germ granule components as potential therapeutic targets? So I'll leave you with that, and I think the last person is Dr. Yin.
Okay, last or least, I've heard you have no slides.
I have no slides.
I mean, that's courage.
or stupidity, I'm not sure. So, my name is Vut Yin. I want to welcome everybody. This is the most important organ in your body. You see, it's responsible for delivering oxygen-rich blood to every single cell and every tissue. And yet, for an organ that is so critical to life, it actually does a really poor job of repairing itself after an injury. So for example, if you or I were to suffer a heart attack, our hearts will actually seal that injury with a very rigid, inflexible scar tissue. Now the problem is that scar tissue is a major impediment. It actually prevents the heart from relaxing and contracting. So you can imagine that if that initial damage was extensive enough, it could actually be the first domino that can ultimately lead to heart failure. But what if we could change that? What would you say if I told you that everyone sitting here today actually has the capacity to regenerate heart muscle, and that instruction manual to achieve that remarkable biological process is actually already written in each one of our cells? The challenge is we need to decode that instruction manual. We need to identify the genetic switches that need to get turned on and the genetic switches that need to get turned off. And it's through that combination that we can ultimately unleash our own body's ability to repair itself. So how are we going to tackle this incredibly important question? Well, much like my colleagues, I operate under the premise that, and to quote Leslie Orgo, nature is a heck of a lot smarter than I am. Because the simple fact is, nature has actually perfected the process of tissue regeneration. And the heart is no different. So a strategy that we adopt is very simple. Identify the system that regenerates remarkably well, perturb that system, learn from that system, and apply that knowledge for human health. Well, that system of choice is the zebrafish. Now, this is really an amazing organism. You can poke it. You can freeze a portion of the heart. Heck, we even cut off a portion of the zebrafish heart. And these animals will actually make new heart muscle, get rid of that bad scar tissue, and the heart fully recovers its function. But the interesting thing is that genetic toolkit, that the zebrafish deploys, we have that as well. We have those same circuits because nature has perfected that process. So that's a major goal in my lab. We want to use the zebrafish to understand both the genetic and the cellular underpinnings of how to regenerate heart muscle. But we can also do something else. You see, we can use the zebrafish as a platform to screen small molecules with the idea that if we identify a small molecule that actually enhances the rate of regeneration in the zebrafish, well, that tells us that that small molecule is manipulating those key genetic switches that allows regeneration to occur. So we can take that particular small molecule and we begin testing it in a mammalian model. And when it comes to heart regeneration, that mammalian model is the adult mouse. Well, we've taken that approach, and as Peter alluded to, one of the remarkable molecules that came out of that screen is MSI 1436. Now, MSI 1436 does some remarkable things. When we administer this compound to an adult mouse after a heart attack, these animals survive longer. They reduce that bad scar tissue. It actually stimulates regeneration of heart muscle. But perhaps the most compelling finding is that it actually improves heart function. So what does this tell us? MSI 1436 is a compound that stimulates regeneration in the zebrafish and in the mouse. Two animal systems that last shared a common ancestor about 400 million years ago. So it tells us that the genetic circuitry for heart regeneration is absolutely conserved during evolution. So what are we doing now? We're using MSI as a critical tool to understand those additional switches, those additional toggles that we need to manipulate in order to fully release our own ability to repair our injured hearts. So if you want to learn more about some of the unique applications of MSI 1436, or if you want to learn more about our research program, how we can regenerate the most important organ in your body, please come see me. Thank you.
Thank you very much. Well, I mean, obviously, we have a discussion. We will have a discussion about that. It's the kidney. I mean, what a wonderful faculty. It's an honor to be president of an institution with such a faculty. It's great to learn from you. I had the pleasure to sit down with you and talk about your programs and what you're doing. And I'm learning, and it will be great to do this together. Now, if we go on, one of the challenges here is not the excellence, but is the focus, and I'll come to this in a minute. The challenges for the faculty and for the institution is recruitment of senior scientists. We have an ad out, and this will be very important over the next couple of months, recruitment of new research groups. It's very obvious that we need your support as well, because this is not something we can do all by ourselves. Here we need grants, but in addition to that, we need more resources. Excellence, I already said, is not the problem. Focus is the problem, because some of you, and I can understand this, are a little bit overwhelmed. But it's the same for us. You know, I mean, these are research areas which we have to bring together. I mean, we have to be sharp. And Eric, this was really not discussed between us. But I promise you, we have to go there together. This is Lukas Granach. We both had the numbers here right. It's 1546. It hangs in the National Gallery in Berlin. And I promise you, I've been standing in front of this often, and we'll go there together. You can call it the Fountain of Youth, but actually you could call it also Aging and Regeneration 2018 MDIBL. It's a very nice example. We could talk for about, well, let's say 35 minutes about that. And you can see here aging, you can see here regeneration, you can see here fun. Aging is a little bit more of a problem. But the question is focus. And focus is here. That's not my idea. When we discussed this, we said, well, it's exactly the line where aging and regeneration interact. And you've seen this throughout the presentations, that this could be one focus. Focus does not mean in science that everybody has to do the same and always link it to this one line. But it's a very nice example of what we can achieve in an academic campus when we contribute to this discussion, because, as Eric mentioned, this is not done very often. Everybody is getting positive results, then they publish and they get the next round of positive results. But to talk about why this is important, and especially about these things, and I think this, I'm looking forward to this. Next of the 4 pillars here is a fellows program. I leave out the word here, visiting scientist program, because we have a complex history over the last 10 years, and some of you have an understanding what a visiting scientist program is. We need a new visiting scientist program, and I'd like to call this a fellows program. But basically it's the same. MDIBL, we as a faculty and the staff, we are hosting international science. We'd like to have spirited discussions, novel ideas, new collaborations, and good teaching. But as all good hosts, we also have criteria for our guests. How long do they stay? Do they contribute, and do they do the dishes? What do they cost? What do they bring with them? We are not looking for presents, but support would be nice. Where do they work, and where do they sleep? So these are the questions we are facing now, and most of you know that this is challenging. So what I think we should do is we will have a program where we have 2 sorts of visiting scientists. One are the fellows of MDIBL. These are scientists who come back every year and who have been, together with the faculty, the backbone of MDIBL. This means with such a program, we are widening our faculty, so to speak, at MDIBL. We need excellence. We have a stability, not every year, but they have to be here very often. They bring their own crew, and they contribute by teaching. We have visiting scientists. They are also excellent. We will have collaborators here, and they also teach. But we do not need the stability from them. The recruitment will be difficult because we have visiting scientists here. The faculty has visiting collaborators here, but we have to do more PR on that. We need lab space, and I've been talking with the faculty about that. We need methods, preparations, but most of all, we need housing, and the campus development program will be an essential part of that. But you understand, we have then a fellows program where we can ask for your support and for support from donors, not for cottages, but for a program, for a scientific program which we need for the development of MDIBL. Education. I will be very brief here. Jane Disney and her crew, they have done a wonderful job, and we will have lots of discussions about the students' courses. We want high standards, we want enthusiasm, and I've already alluded to that. We have a lot of courses at the moment. We need a discussion, which courses do we actually need? There are financial aspects, but there are also aspects which courses contribute to here and to here. And then we have conferences. And you've already heard from Vicky about her conference and how wonderful this will be. And I think now, Eric, it's your turn.
Thanks. I just wanted to take a couple of minutes. In fact, I even wrote some stuff out so that I wouldn't babble for too long. Just a quick overview of the Regen and Aging courses. This is a flyer for my Aging course, which starts this weekend. This came about sort of a follow-up to Voot's regeneration course, and from the COBRI to sort of synergize and bring together the skills and the type of research that the COBRI is meant to fund, and bring courses that reflect that, and also to bring scientists here. You know, we're a small group, and we acknowledge that, but we have a place that people love to come to, especially in the summertime, and so. It's actually not too hard to recruit people to get them out here for this. And for the Aging course, I can say we're sort of leveraging a legacy. If you've heard of Larry Ellison, maybe, Oracle guy, for 15 years, he funded the aging community very well. He gave New Scholar Awards, Senior Scholar Awards, and he funded a course. Very nice course for students or for postdocs and grad students trying to get into aging research. And this became a fundamental thing in the field, really important. Larry Ellison might be a little bit mercurial. I hope he's not out there right now. But he pulled his funding. He gave $500 million, and then in about 2013, 2014, it stopped. Now, this could also be because things like Calico, Google's Calico, were coming down the pike, and other aging biotechnology resources and funding. So there was this expectation in the community, but nobody picked up on it. It was just great timing to be here in a place where we could actually sort of pick this back up, improve it, and make something, frankly, that would be an opportunity for the highest caliber of new scientists in the field to sort of network and get in, hear what is happening in all the major model systems, hear from the best scientists, be trained by people who have cutting technology that they've developed in their own lab. The one thing I was really nervous about was that Larry Ellison fully funded that other one, and we had to charge tuition. And I thought, is anybody in the community going to come here and pay money? Several $1,000s. This is an expensive course. I was really pleasantly surprised that they did. We also got some very important seed funding from the Glenn Foundation, whose interest is in extending the healthy years of life. They gave us $20,000, which we were able to give back to some of the students in the way of tuition remission. So I just want to tell you a little bit about some of the students that are coming here. It's graduate students and postdocs, as I mentioned, future leaders. We have several postdocs from Harvard Medical School, National Institute of Aging, Kogod Aging Center at the Mayo Clinic, INSERM, which is France's NIH. We have other talented folks coming from other places, Australia, Denmark, across this country, of course. We have a brilliant physicist who's with the Weizmann Institute in Israel who's switching fields. He's already got himself placed into a couple of very high-profile biology labs, and he wants to become an aging researcher. So I have a special appreciation for that coming from a different field. This year, David Sinclair, whose name is one of them you can see up here, one of our invited lecturers. He's not only is he an aging biologist at Harvard, but he's also an entrepreneur, a very successful one, and he's now affiliated with a country called Life Biosciences. They just launched in the last month or so. Keep an eye there. So they'll be giving Google's Calico, presumably, a real run for their money. They've offered to give the course $100,000 each year. So we can achieve our dream, really, of just focusing on making it a competitive process to get the best of the best students in and to become something of a mecca here. Now, I really have to thank that getting an agreement for $100,000 for a course like that is kind of a big deal. It's our first big corporate sponsorship, and I just want to thank several people who helped me in these sort of new waters. Jerry Bowers, Judy Sproul, Claudine Lurvy, Jane Disney, Anna Farrell all helped secure that, and I just want to thank them so much for that. Also, thanks in large part to Jane Disney, Vood and I have and from her pushing us, we have a proposal, an innovation proposal in at the NIH. It's an innovation grant called an IPERT, and what this would do, not just fund a course like this, but it'll actually help us set up a community. It's really meant to create a new area or build on an underdeveloped area. So we're really crossing our fingers about that, because this is going to be the first time we really see a merging of aging and regenerative biology. And if you think that they should be talking, you're right, but they are fundamentally different. They are fundamentally different. There are different requirements for accessing each of these pathways, or the myriad of pathways involved in slowing aging or in regeneration. So what we're looking to do is combining the Regen Aging course into a 21st century course. And let me just give you a tiny, super tiny example. If you cut a mouse, give it a little cut, it heals. If you have a mouse that's on dietary restriction, it lives longer, it lives healthier, doesn't get cancer, which is usually what lab mice die of. If you have it dietarily restricted and you give it a little cut, it will heal, but it heals more slowly. Now take that same animal, dietarily restricted, give it a cut, now feed it full. It's used to being on dietary restriction, now it's receiving full nutrients, it heals faster than the control. That simple little concept is going to be key and fundamental. Remember how on my screen I had growth and reproduction on one side and energy conservation and maintenance on the other. It's going to take a concerted effort to marry these fields, but that's hugely important because in addition to personalized medicine, these are the big ways going forward in medicine. Whether you believe it or not, I'm telling you here, aging and regeneration, that's where it is. That's where it's going to be. How does something like Voot's regeneration drug, how will that interact with our anti-aging strategies? So in this new course that we're proposing to the NIA and others who have expressed interest, one of the things we'll be doing is a nexus experiment. So this is going to be something where we purposely marry these technologies, these areas, and we figure out how to put them together, because at some point in the not too distant future, medical doctors are going to need to know this information. We need the basic research to inform how you would put these sorts of things together. What is the best way to maximize the benefits? What's the best way to time these things out? So I'm very excited that we have these opportunities. So this is our new big partner and our other nonprofit philanthropic partner. And very excited it's going to get going here in the next few days. And there'll be several public lectures that everybody can participate in and should be digestible to just about everyone. So I would encourage you to come for those lectures if you can and get a chance to hear from those scientists.
I don't think we need to explain the word enthusiasm again. However, this is really very important because you see here, this is not only world-famous lecturers. Actually, this course has everybody on it at the moment who is in the field important. But it also has laboratory instructors. And it has here MDIBL together with the Jackson Laboratory. So this mixture of lectures, labs, if he could have, Eric, this course not only for 1 week, but for 2.5 months, that would be the visiting scientist program for aging on a very high quality. So this is where we want to go. However, we also, in building on things like that, we'd like to have Acadia conferences on MDI. They will start with MDIBL. The Jackson wants to participate, and the role model is Keystone. And Keystone mixes natural beauty, outdoors activities with excellent science. And people come from all over the world, like for Eric's and for Vicky's course, and I also have one in the fall. And we have everything on this island. So we will use natural beauty, not downplay it and say, well, that's only good for the summer, but use it for these courses. So we will have international conferences of the highest scientific quality, and you have just seen one. All the qualities of MDIBL make it an ideal meeting place, intellectual excellence and outdoors activities. And last not least, Keystone makes money out of these meetings. They are seriously funded. Every meeting, Keystone makes something like 250,000. And since we have most of the things already here, I think we could use this. This has to do with funding through pharmaceutical companies. It's basic research, but as Eric just mentioned, they're interested in that. I started to talk to them. This is where I have lots of connections, and they are interested. Bayer, for instance, has said next year, cortisone, aldosterone, an Acadia conference here on the island, they would be very much interested. Last but not least, biotechnology. Biotechnology is really hard. We have started this. We have now the new building. We have to do biotechnology. You have been told that I have funded companies, I have learned my lesson the hard way. You ask my wife. Now, this is not easy. And one of the things which is very important, scientists are not entrepreneurs. Some of us think of new ideas and they think of a lot of money, and that's all fine. This is why we are scientists. But to run a company, to make an idea work in the real world is something very different. Sometimes entrepreneurs dream late at night that they could be researchers. It's different. So we are not that. However, I will bring two patent applications to MDIBL. One is inhibition of MCP1. That's an inflammatory drug, and I will not talk about this. But we will also use zebrafish embryos for diagnosis of thrombotic microangiopathy, which is a novel idea how to use these animals in a clinical setting. We planned for 3 companies, zebrafish as disease models, and I already have a company in Germany, mice as disease models, and we will have the part with the zebrafish here on campus. And then the most challenging is that we actually could associate with patient studies in Maine and educate young people, study nurses and scientists, how to participate in studies. This is also income. If we start something like this, the main population, and we understand where sick people are, and they can participate in this study, this is highly paid. Pharmaceutical companies, if you tell them, we have organized this in a way that we can do these studies within a limited amount of time, that's worth a lot. So this is the development of a novel drug, and you have seen this from Wood and from Kevin. We will have zebrafish as disease models here. We will have these, the novel drug inhibitor of inflammatory disease, and we will have both animal studies, but we already passed phase 1-2, so this will be a nice drug to develop. And then this, the clinical research organization, looks dramatically new. I have lots of experience with that. I think we can bring this here, and we can actually help translation medicine to become really at home here. I have to admit, I would not touch this without help. And I'm very lucky that Judy Sproul agreed to step down from her position she has now, and she will start with me this biotechnology activity. We will have courses in translational biomedicine where we can actually teach young people from Maine and from elsewhere what you do when you think you have a patent, where you can look whether this is really patentable. Patent management. We will teach them that when you have a patent, it's not like something you just go there, but then you have to manage it. You have to split it in part. It costs $8,000 to translate it into Japanese, but if you don't have a Japanese translation, it's not worth anything. And these will be taught in these courses. We will have business plans, and we will use these words like exit strategy. Because if you don't have an exit strategy, nobody's going to touch your company at any time. We will have hands-on courses in these companies for company management and development. And as I said, I'm very happy that Judy will do this together with me, and I hope that we can be successful there. So this is it. But now I've started with the roof, and this is the foundation. You can't do this with a serious foundation. So the staff, and here you see all of them, or the different departments, they are vital and crucial for MDIBL, as we all know. So I can start not thanking him enough. We need development, and you will see our goals in a moment. When we talk about a development plan for cottages on campus, this will be a major task. When we have these 4 business units here, the financial system, and Claudine, we have already started talking about that, this will be hard. This will not be done by the crew we have at the moment and not with the computers we have at the moment. So this is not only broadening our basis, but this is really a challenge to achieve in the coming years. Now, these are the challenges for all of us. Successful financial management of all 4 units. We need, and this is what we have to develop now over the summer, we need strategic plans for each of these units. For the faculty, it's recruitment, and then planning, a financial planning, what happens after the COPRI. We are now in the 2nd round, and it is already going down. And when the COPRI is over, we have to have other resources, and we have to plan for that. For the Fellows Program, I've already mentioned this, campus development plan, recruitment. For education, we will not improve this only, but we want to have more students here, and I'd like to introduce the Acadia conferences. And for biotechnology, major challenge in order to bring this really alive here on campus. And we need not only strategic plans, because strategic plans are worse. We need benchmarks. And these benchmarks, we have to look carefully, and we need the board to oversee this. We need your help and support. And I can tell you, without these strategic plans, what my benchmarks are. 1 senior faculty position, 1 junior faculty position, 10+ visiting scientists, 30+ students next year, and the fundraising for 2018 and 2019 should be at least 1.5 million. I know. That is a challenge, and I cannot do this without the faculty, and especially not without the staff. So now this seems to be over, but those of you who are still awake and who have been listening, what did he do with these things here, you know? I mean, were they just there for fun? I mean, I've used all the other building blocks, if you really looked at that. We need them here. We need communication between all these different areas. And even at a place like MDIBL, which is so small, communication is a problem. The faculty, they have organized themselves and they meet every 2 weeks and they sit down and they talk about science. I need communication to understand what's going on, but the communication, we have to talk to each other more to understand what's going on. We are perfectly situated for that. We have a campus, but we have to meet on campus and we have to have lively interactions. It is also not only about coordination, but about cooperation between the different groups. We will only live up to all the expectations in terms of excellence of science if we interact, if we use methods from each other, and we are really together in that, and then we can become much better. So it may look a little bit funny. It looks not like a Greek temple. It looks more like Frank Stella and the Museum of Modern Art. But some of you may think it looks a little bit funny, but this is what science is all about. You know, it's playing with building blocks. It's building something stable, and it's colorful. It may make you smile, and this is what we need. Mark Hanscom said to me, "We need more smiling faces on this campus." Yes, we do. Thank you very much.
certainly like to thank, again, the faculty and Dr. Haller for all the work they did to put in for today. It was really spectacular. I'd like to make one final thank you and one announcement. We'll take 5 minutes before we hit the lunch. First, I'd like to thank all the Board of Trustees. They put in a great deal of work over the past several years in terms of this transition. There's sort of a rowdy group of them over here. Please introduce yourselves to them. They'll be at lunch. They're a remarkable group of people. I continue to be impressed with all of them. One thing that the board recognized over the past year or two is that transition creates a great deal of stress. I've been through it in my employment, and anytime there's a transition at the top, there's a lot of stress underneath. And the board recognized this. We actually, if I'd known about Jim's stress-activated green fluorescent protein construct, you know, we could have tossed it in the water down here and see how many people were glowing bright green. But one of our trustees brought up the issue of retention. We do a lot of recruitment here. We've been doing a lot of recruitment of scientists over the past 10 years, building the group. We now recognize that we have really outstanding talent here, and real poachable talent, if you will. And the Board of Trustees recognizes this. And so what we would like to announce this morning, and Wister, if you'd like to come forward, as well as the faculty would like to come forward. Wister and Martha Morris are just spectacular supporters of MDI Biological Laboratory. In fact, I consider them the absolute heavyweight champions of MDI Biological Laboratory. Wister loves the science. Wister's probably the best scientist on the board. He understands the science. I often see him here with our faculty discussing the intricacies of their work. He's a very impressive individual, as well as Martha. We're announcing the formation of the Morris Scientific Discovery Fund, which for a period of 2 years will provide up to $250,000 for our faculty. And the goal of this fund is to ensure the continued development of the MDI Biological Laboratory faculty by providing short-term pilot project support for research programs that demonstrate a likelihood of successfully competing for federal funding. This is the Morris's effort at retention, and the faculty have a presentation.
So let me just take a few minutes to say a few words. So Wister, as many of you know, is a man of very few words. But I think his actions speak volumes. And it's really been his continued support and generosity that has really empowered the faculty to develop the innovative research programs that you see here today in front of you. I remember asking Wister one day, but a couple weeks ago, I said, Wister, you've had tremendous success in finance investment, but investing in MDIBL is a whole different beast. So what are the metrics? How do you judge whether or not you've been successful in your investment? And in typical Wister fashion, he took a few minutes to think about it. Then he said, the faculty. He said, if my investment and support results in launching the careers of the faculty at MDIBL, that would be quite the achievement. So I hope, Wister, you can look at the faculty. You clearly know what we do. I think you recognize the achievements that we've been able to do. And I hope you take pride and ownership of having an instrumental hand in elevating what we do here. And on behalf of the faculty, and as a small token of our gratitude, we'd like to present this to you and Martha for your continued support and generosity of us all. Thank you very much.
I'm going to be very brief, but let me just say a few words about why I am so optimistic. Let me get out my notes. I mean, it's very gratifying to hear what you had to say, but it's even more gratifying to hear your progress and see your results that you put up here just now. And let me give you and that makes me alone very optimistic for the future of this organization. Let me give you a second line of reasoning. We had a finance committee earlier in the week, and what you see on the balance sheet for 6/30 is $8.2 million cash and CDs, $2 million grants receivable, and $6.7 million of investments in the endowment. That totals $17 million. Against that, we have a $4 million bond due from now till 2036. So we have a long runway. On the income statement, we have depreciation of the buildings and the equipment of annually $1.4 million. That's a non-cash cost. So even though the income statement shows a loss, we're positive on a cash flow basis. And I'd like everybody here to think about getting a copy of the 26-page annual report audited, and maybe have our Chief Financial Officer Extraordinaire, Claudine Lurvy, give you all a brown bag lunch opportunity to learn about it and hear her presentation. Let me return to the subject of biology and the medical science that we do here. There's a book, The Youngest Science, by a Dr. Lewis Thomas, who graduated med school in 1938. And he went to work at Johns Hopkins, and he said they had 3 drugs. They had, at that time, they had emetics that clean you out, they had palliatives, which put you under, and they had placebos. My point is, that in that 80 years, there's been really incredible progress made. And you guys are working at a time now, and girls, working at a time when this is the golden age of medical discovery. So it's really your turn to do this, and I wish you every success. And I'm glad to play a small part in it.
Thank you, Richard. Thank you. Thanks again. We will adjourn for lunch.